Okay, we're into the last chapter of Unit 1. This is all about microscopes, and we're mainly going to focus on light microscopes because that is what we're going to use in our lab. So, this is somewhat like our light microscope. We'll talk all about the pieces of it, but it is what we call a compound microscope that has two lenses, and the light actually shines up through our sample. We're also going to talk a little bit about kind of the scale of organisms in terms of what we can and what we can't see. Obviously, for microbiology, everything we're going to look at is microscopic, but there are some things that are so small, like viruses, that we can't actually even see them with our light microscope. So we'll talk a little bit about that too. So, like I said, mainly talking about light microscopes, but also about why they're really important and useful. They're still super useful clinically to figure out what kind of pathogenic microbes might be causing disease in a patient. We'll look at different types of microbial cells, and we'll do this a lot in the lab. We'll spend a lot of time looking at stuff under the microscope in the first few labs. And then we'll talk about kind of what type of microscope might be used for a specific type of image. There's kind of three that we're focusing on: light microscopes, electron microscopes, and then fluorescent microscopes that are really useful for us. Okay, so let's start with a case study here. This one we have Mariana and Julio, both 8 and 11, and they tell their mom that they can't see right and that their faces are starting to feel numb. So this occurred about an hour after they'd eaten some hot dogs with this chili sauce on it. So, of course, here's Sonic. He loves chili dogs, right? And this wasn't home prepared. This was commercially canned chili sauce. So they go to the urgent care, and they're found to have normal vital signs in terms of pulse and things like that, but their pupils are fixed in this dilation, and they have what we call symmetrical weakness, which is equal weakness on their left and right side. So you can see kind of a representative image of this here. Kind of you can see the muscles drooping, the eyelids drooping equally on both sides, and then these fixed dilated pupils. Scarily, this weakness progresses into paralysis and difficulty breathing. So they go to the ER and they are put on a ventilator to actually help them breathe. So at this point, the doctors would have asked, what were they doing? What did they get into? What did they eat? And that eating is probably what tips them off. But in this case, we suspect that there's some sort of toxin, paralytic toxin that's in there. And a common one is botulinum toxin. And so they do a test using what we call a fluorescently labeled antibody. So an antibody is an immune molecule. You make antibodies against microbes. And we can use that because antibodies are super specific. They target specific molecular shapes. So we can make an antibody that recognizes this botulinum toxin, and then we can attach a fluorescent dye. Fluorescence means glowing. So if that antibody binds to something, we see something that glows. In this case, this image, we see glowing bacteria because the antibody is binding to them. In this case, it would be to the toxin itself. And when we see that positive test there, it's really specific. This antibody is super specific to the botulinum toxin. So they're diagnosed with botulism, which is a paralytic illness. And it comes from contaminated foods most often that are contaminated with Clostridium botulinum, which is a bacterium that makes this botulinum toxin. Because it's a paralytic toxin, that ventilator is going to help them breathe while their body fights that toxin. So the treatment is just to kind of wait for it. After a few days, they're removed from the ventilator, and with physical therapy, they're able to make a full recovery. We're, of course, going to then investigate the source, and because they ate some canned food, we're going to go investigate the canning factory, and that's where they find evidence of the bacteria there. So Clostridium botulinum is interesting. It makes these kind of club-shaped spores. So this part is the dividing bacteria, but then it makes this protective, what we call endospore. And these endospores are super resistant. They resist heat, so even if you boil something, it doesn't destroy these endospores, and they can live without oxygen. So botulism and canning are a problem because canning is designed to preserve food, right? You heat it up and you seal it, sealing out the oxygen. Hopefully nothing can grow in there. Well, these botulism spores can survive that. And this causes a low number of cases in the United States because we're aware of it and we test for it, but about 24 cases a year, and it's very scary. I also saw some instances where honey can also have Clostridium botulinum in them, and so sometimes people feed their babies honey, and it causes what's called floppy baby syndrome, which is basically paralysis. So that is something new parents should be aware of, right? But noticing those signs is important. So if you looked under a microscope at some of those cans, you would see these specific bacteria, and by the shape you suspect Clostridium botulinum. Then they also use that antibody test for the toxin itself. And there's actually a specific stain that we can use to find this as well. So I'm going to ask you a question. Who knows what botulism toxin is also used for? Think about it, pause the video. You may or may not know this. That's right, Botox. So, this is, I don't know, scary, I guess, if you're not into injecting bacterial toxins into yourself. But along the way, somebody realized that, hey, this paralytic botulism toxin, botulinum toxin, it can paralyze facial muscles, and that prevents wrinkles. So literally, they grow the bacteria and purify the toxin from it, and then whoever is doing it, a dermatologist or cosmetic surgeon, can inject the toxin into the facial muscles, and it literally paralyzes them, and your wrinkles go away. I personally think this is a little extreme. You're all beautiful just the way you are, particularly without bacterial toxins injected into your face. But everybody has the right to choose what they do. But for me, this is a little bit much. Okay. So in 3.1, we're going to talk about observing microbes. And this is really important because, as we saw historically, we didn't know microbes even existed until we had microscopes, because by definition, they're too small to see. So we'll talk about how a microscope works, talk about magnification and resolution of images, and we'll learn about a few different types of microscopy. We'll focus just on a few specific ones. Okay, so I want to show a reason why a microscope is useful. Our eyes are super powerful, right? We have the ability to observe our world and notice patterns and things that are going on. But we have a certain kind of limit to what we can see, right? So in this image here, you see two vials. One of them is clear and one of them is cloudy white. So this is cerebrospinal fluid. So that is literally the fluid that is in your spinal column, basically, and goes up and coats the brain in the meningeal area. So it's really critical, because of the importance of our spinal column and our brain, that there are no bacteria in this fluid, right? It is hopefully a sterile fluid. In this case, we're observing this cloudy cerebrospinal fluid that we can observe. This cloudiness is due to white blood cells. So this is an indication that there's an infection going on, right? We can't actually see that there's bacteria in there, but we can see that something is wrong because it's this white cloudy color. If we take this and put it under a microscope, we have the ability to resolve the individual bacteria. We can see the bacteria that are in there, these little pairs of coccus-shaped or circular-shaped, spherical-shaped bacteria here. So over here, we're able to detect a problem, but over here under the microscope, we can actually resolve the cause of this. And just seeing this shape and this arrangement of bacteria gives us a clue as to what it is and what we should prescribe to treat it. So we need to talk about the definition, right? We talked about microbes being microscopic. Well, microscopic literally means requiring a microscope to be seen. And a microscope does magnification. Magnification increases the apparent size of an image. But magnification is not everything. If you've ever taken like a picture you took on your cell phone and blown it up, right? Tried to zoom in, you might notice that it doesn't necessarily get clearer. That's because there is a limit called resolution. Resolution is the smallest distance at which two objects can be separated or distinguished. I'll show you a picture in just a moment. Really what this comes down to in your camera, in your cell phone, is the pixels, right? How many pixels does your camera have, right? That resolution is the limit. You can magnify all you want, but it doesn't increase resolution. You have to have a better camera that has higher resolution to do that. Our eyes, believe it or not, are kind of like a camera. On the inside of our eyes, we have a set of cells, rods and cones. We're not going to get into this too much, but they are arranged at a specific distance. And this is literally the resolution of our eye. In kind of the center of our vision, we have these cones arranged very, very closely. That's where our highest resolution is. That's why we can see best in our central vision. And then around the outside, they get a little bit more spaced out. That's why your peripheral vision is not as clear as that central vision. And what happens, right? Your eye, it's got a lens in it. And that lens takes in light that's reflecting off whatever you're looking at, this dapper gentleman here perhaps, and the light goes in and it hits that lens and gets shot onto these nerves back here, these receptors, and the brain gets that signal through the optic nerve and then interprets that. Okay, so that's our eye, right? The resolution of our eye is about 150 micrometers, and that really doesn't mean much to us. We'll kind of see what that means more in the lab. But an eagle's eye actually has a much higher resolution. It is 8x times better than our eye. So that's why we call people eagle-eyed if they really have good vision, right? Because an eagle actually has cells that are 8 times closer together here, so it can resolve much better than we can. So right here's an example of resolution. High resolution, right? We can distinguish between two different things much smaller, so like the individual hairs here. Whereas at low resolution, there's in this case, fewer pixels in here, right, per area, and we can't resolve those hairs there. We can make out the broad structures, but even if we zoomed this in more, it would just get blurrier and blurrier, right? The things that we talk about in microbiology, they vary in size, right? And we have things that range from rather large microbes, I'm going to say. They're still very small, 33 micrometers, so below the resolution of our eye, right? Let's talk about units for a moment. We'll do a whole lab on units, but in science, we use the metric system, which is actually very convenient because everything is by a power of 10, right? So we have a unit of measure called the meter. It's about 3 feet, and then thousandth of a meter is what we call a millimeter. So 1 millimeter is a thousandth of a meter. In scientific notation, that's 10 to the minus 3 because there's 3 zeros here. So 1,000th of a meter. We also have a unit called a micrometer. This little mu here is the Greek letter that we use for micro. 1 micrometer is 1 millionth of a meter, so 1,000 times smaller than a millimeter. 10 to the negative 6 in scientific notation. You've probably heard of nanotechnology. That's things that are on the nanoscale. A nanometer is 1 billionth of a meter. And then there's even pico, which is a trillionth, 10 to the negative 12 down there. This is kind of the scale we're going to work on with our light microscopes here. So you might not have this visual image in you, right? But your red blood cells in your blood, they're about 8 micrometers. So this eukaryotic parasite, Trypanosoma brucei, is about 33 micrometers. It's bigger than a red blood cell. Then we jump down to something like bacteria, and this is Bacillus megaterium, and it's named mega because it's big. It's a big bacteria. It's about 4 micrometers. E. coli, 2.5 micrometers. So much smaller than these eukaryotic cells. Staphylococcus aureus, which can cause skin infections, 0.9 micrometers across. And then we get down to the viruses. This is a big virus, herpes virus, 0.2 micrometers. That's pretty big for a virus. This is getting beyond the limit of what we can see even with a light microscope. You have to go down to an electron microscope. So what do I mean, different types of microscopes? We're not going to go too deep into optics, but we will talk a bit about it. A light microscope literally uses light shining through an object to give us an image. A lot of times this requires us to stain the sample to give us some contrast, and we'll talk about what that means. There's a couple different types. We're just going to use general, what we call bright field light microscopy. There's a special type of light microscopy called phase contrast that can help kind of bring out three-dimensional structures. The instructor demo scope up on the front bench in our lab has one of these filters on it, so I'll show you that in lab. Then there's fluorescence microscopy. This uses light, but it uses special glowing light that fluoresces, like when we talked about that toxin, the antibody labeled with a fluorophore, that can glow. To do this, you need special dyes and equipment. Then if we want to get really small, we have what are called electron microscopes. There's one called a scanning electron microscope. This gives surface detail. It uses basically, it's kind of like a phonograph needle or record player needle, and it scans over the top of a surface. But instead of using light, it uses electrons, which have a much smaller wavelength, so we can get higher resolution of objects. So you'll notice this is down to 10 micrometers. And then we have transmission electron microscopy. This shoots electrons through a thin section of it. So the really cool like 3D images of small stuff, generally scanning electron microscopy. Transmission electron microscopy looks like this. It looks like kind of like light microscopy, but with super high resolution. So if we put this into perspective, right? The human eye, we can see a millimeter down to about what we say, 125 micrometers. So there's some big eukaryotic parasite things that we can see just, right? Past that point, we use light microscopes, and we get into looking at bacteria. And then below that, if we want super detailed resolution, here's an E. coli cell. If we look at it here under an electron microscope, we can get really fine detail. And we can even get down into subcellular pieces. So if we looked inside the cell, we could see this is a mRNA here, and these are ribosomes making little chains of proteins coming off of it. So subcellular, inside that cell, we can see with electron microscopy. But at some point, it's too small even for that. And beyond that, we use a really special technique called X-ray crystallography. This is what Rosalind Franklin did to figure out this part of the structure of DNA. And it uses X-rays that bounce off atoms, and then you use a computer basically to reconstruct the shape. This is for like molecules. So this little blob here, this ribosome, this is kind of the molecular shape of it, and it gets kind of weird at this point. We're going to focus mainly in the light microscopy section because we're mainly going to look at bacteria, and we'll also look at some eukaryotes. For viruses, you need electron microscopes. So what does this mean for us? Well, part of identifying a pathogen, right, is seeing it. So for us, light microscopy is a useful technique to do this. We can identify the 3 common shapes of bacteria. We have rods, which are rod-shaped. They're also called bacilli. So this is a bacillus here. We have spiral-shaped bacteria called spirochetes. These are really skinny and small and hard to see with our light microscopes. They're very small. We saw an example earlier in chapter 2 of Trypanosoma pallidum, which caused syphilis. That's caused by a spirochete bacteria. And we have the cocci shape, the coccus shape, which is a sphere. And this is a chain of these spheres. So you may have heard of streptococcus. This is probably what that's an image of. So these are light micrographs of that. So these are light microscopes. They've been stained to give us better contrast here. If we look at the same things under a scanning electron microscope, we can see much more detail, right? This thing here, now it's this long shape here, and we can see the 3 dimensions, super high resolution, right? This color that's been added here, that's all fake. Electron microscopes don't see in color because they use electrons, which don't have colors. The spirochetes, you can really see the spiral shaped here, and then that coccus chain, you can see the spherical bacteria all chained together here. So these are the 3 shapes: rods or bacilli, spirochetes, and cocci. All right, so detection, right? We gotta be able to determine the presence of an object, but to really see it, right, we need magnification, so increasing the apparent size of an image. But we also need resolution to tell the difference between 2 small points. Magnification is great, but it doesn't help if you don't have increased resolution as well. Eukaryotes, they're generally big. We can see them quite well under the light microscope. We'll even look at some of our own cheek cells in the lab. We're eukaryotes. So bacteria are much smaller. Bacteria, it's hard to see inside the cell and what's in there, but we can see the shapes. And there are different types of microscopes that are required for different things. Generally, when you get smaller, you got to go to electron microscopes.